Well, fine, children. Good morning, all. Good morning, children.
Morning, sir.
Okay, fine. Okay, let others join. We'll start a revision of the second chapter. And the second chapter we have seen till about the andrecium and the gynecium, and we are focused on the structure, and we are focused on the events, and we are focused on microsporogenesis and pollen grain formation till their development, and we saw about megasporogenesis until the embryo sac development. Is there any doubt in it, children? Any doubt in it?
No, sir.
Okay, so based on that, children, today this PPT I'm going to present, which I already have shared you. In this PPT, it's just for your revision purpose alone and only. And you may say, so I want notes for it. Where to search? You can search the notes from here. So you know that sexual reproduction in flowering plants, where we are not going to discuss about thallophyta, bryophyta, pteridophyta, and gymnosperm. We're going to discuss only about an angiosperm, and which is unique for monocot and dicot plants. So all flowering plants they show the sexual mode of reproduction, and flowers are the site of sexual reproduction. Among the flowers, which is the essential part required for the reproduction?
Andresium and Ganesh.
Andrecium and gynecium, where andrecium represents the male part and gynecium represents the female part. So several hormonal and structural changes results in differentiation of floral primordium. Inflorescence, arrangement of flowers on the floral buds is called as the inflorescence, where we have learned about racemose and cymose, but nothing to do with your grade 12 now. Now, the flower, we're going to see about two essential part, andrecium and gynecium, because we know that sepals called as the calyx, petals called as the corolla. These are the non-essential part of a flower, which aids in reproduction by some modifications. But when we look about andrecium and gynecium, they are definitely they required for a reproduction. So we look about a male flower. Of course, with the male flower, they have a stamen, and the number and length of each and every stamen varies according to the species. So it is actually a bilobed anther. So we have to discuss about the structure of an andrecium. We saw that they have an anther and they have a filament and they have the basal stalk attached to the floral axis or to the thalamus. Now we will look about them. The important part is filament and anther. So you all know that it is actually a completely a bilobed anther. I just change this somewhere else because this may be a disturbance for us. I don't know where to play. Okay, we'll place it afterwards. Now we look about here. So when look about a flower, first one is the filament, the long stalk which connects the anther and the basal stalk is called as the filament or the filament which we talk about. And next when we talk about here, we're going to talk about a bilobed anther and connected by an adhesives. And when we look about a bilobed anther, when seeing a cross section, I can see pollen sacs or microsporangium. So when you look about this, the terminal end is actually a bilobed anther and have two theca, since they are called dithecous. And each and every theca will be having, when you look about an young stage, when we look about them, they may have the microsporangium or we can say the pollen sac. So the anther is a tetragonal structure having four microsporangium at the corners. The microsporangia develop into a pollen sac where we have a sporogenous tissue which differentiates into a cell which is the microspore mother cell. Now here when you look the microsporangium or the pollen sac, they have three protective layer, one nutritive layer and one mother cell. So we look about a microsporangium. They have four walls: epidermis, endothecum, middle layers. Epidermis, endothecum, single layer, but 2 to 3 layer thicker is the middle layer. Next, we have the tapetum. Tapetum will nourish the developing microspore mother cell. Now, I'll ask you a question. Name the cell which will nourish the embryo sac in a megasporangium. It starts with N.
Usually.
Nucellus, very good. Nucellus will nourish the embryo sac. So the outer three layers, that is the epidermis, endothecum, and middle layer, they will give you the protection. And the tapetum nucleus is very important for your examination. The tapetum is actually the nourishing tissue. They have dense cytoplasm, and the nucleus is generally they have more than one nucleus. When they are young, the sporogenous tissue, and they mature into a microspore mother cells. So from the microspore mother cell or pollen mother cell, undergo meiosis 1 to form a microspore dyad, undergoes meiosis 2 to form a microspore tetrad, and all the microspore are functional. Only one microspore is functional in case of microsporogenesis.
All are same.
All microspores are functional, and each and every microspore they give rise to a pollen grain. That is called as microsporogenesis. The formation of microspores from microspore mother cell is called as microsporogenesis. The cell division that is involved in microsporogenesis is meiosis. After microsporogenesis, from every microspore, the pollen grain has to be formed. For the formation of pollen grain, some amount of mitotic division and mitotic differentiation. I'm saying two different words. Mitotic division, mitotic differentiation takes place. So from the anther, we get the pollen grains from the microsporangium. Now we'll look about the pollen grains. Pollen grain is haploid, but from there, what will occur? We have two types of cell here. One which are large one. The larger one is called as vegetative cell. Smaller one is called as the generative cell. And when you look about the outer layer, it is made up of exine. Inner layer is called as the intine. Exine is made up of and substance. What is the substance, children?
Sporopollen.
Sporopollen, which enables the pollen grains to be preserved as fossils. Why? Because acids, alkali, and any other are even they can withstand high temperature and pressure. And acids and alkali cannot destroy, so pollen grain can be preserved as fossil. But when we look about here, there are some areas sporopollen is absent. What is the area called as?
Germ pores.
Germ pore. So through germ pore, some tube arises. What is the tube called as?
Pollen tube.
Pollen tube formation. Very good. I'm so happy. So when you look about the hardest outer layer is made up, or is exine is made up of sporopollen, a highly resistant organic matter. It can withstand high temperature, strong acids and alkali. So enzymes cannot destroy sporopollen till now it is known. And it is absent in an area called germ pore. So pollen grain can be preserved as fossil because of sporopollen. And next we'll look the intine layer. It is made up of cellulose and callus. Pollen tube, though they come from the germ pore, but actual layer from there, the pollen grain arises from the intine layer. Now a question I'm going to shoot off. We have the vegetative cell and we have the generative cell. Generative cell undergoes mitosis to form two different cell. What are they? Cell or a type of a haploid cell. What is that?
Male gametes 1 and 2.
Yeah, the first male gamete and the second male gamete. So here it is mitotic division. The vegetative cells undergoes mitotic differentiation. That means cell won't increase the number. Some differentiation is occurring. So what does the vegetative cells transforms into?
Tube nucleus.
Tube nucleus. Now I'll ask you a question. Tube nucleus will be there always in the tip of a pollen tube, children. In the synergids, we have an apparatus called filiform apparatus. So when teaching you about filiform apparatus, remember that vegetative cell undergoes mitotic differentiation to form a tube nucleus. Generative cell undergoes mitotic division to form two male gametes, first and the second male gametes. Vegetative cells is bigger and has a large abundant food reserve and a large irregular shaped nucleus. And genetic cell is just very a small cell. And of course, when you look, they will float on the cytoplasm of the vegetative cell. And what is two-celled, three-celled? Two-celled means vegetative, generative together we're going to call two-celled. Most of the angiosperms, pollen grains will be shed at a two-celled condition. What is three-celled condition? Where the vegetative cells will be transformed to tube nucleus, generative cell will be transformed to two male gametes. That is called as Three-celled condition. Don't get confused. There will be two vegetative, one generative, or don't get confused. There are two generative, one vegetative. It is fixed that they have only one vegetative and one generative. Based on the mitotic division and differentiation, if mitotic division and differentiation occurs, then it is three-celled condition. So gender cell give rise to two male gamete that pollen grains are shed at three-celled condition. In three-celled means two male gamete and one transformed tube nucleus from the vegetative cell. Now, when you look about them, the pollen grains have a viability. Pollen grains of some cereals is 30 minutes, so it varies from one to another. But I want to highlight an important thing that these pollen grains are edible and they're rich in nutrients. You can see there are some products like biscuits and all this they have made from the pollen grains. Pollen tablets and syrups increase the performance of athletes and race horses, and they are stored under liquid nitrogen. Always one mark, and for a new pattern MCQs will be what is the temperature of liquid nitrogen? Minus 196 degrees Celsius. Later, when time permits, when I'm teaching you the practicals, I have created a new classroom for practicals where you can. I have given you the invite. You can join for practicals. Inform Vidya, Akshat, and yeah, these two people who have joined and integrated to come and join in the classes where practical will be conducted. But I'll tell you my experience where I have worked with the buffalo testers under liquid nitrogen. I did some research work, and the product was also got patented. It's my own innovated work. I'll just share with you. And because of that only, I got Young Scientist Award. I'll share with you afterwards. So pollen grains, even they means of some plants like Parthenium carrot grass are allergic for some people. It leads to chronic respiratory disorder like asthma, bronchitis. This time for the mock 3 or a mock 4, I asked the students about this Parthenium only, but they're not able to answer. Then I told them the page number, then they identify and they have learned, but they're not able to write in the examination. Now, that's all about the male part. Now we are going for the gynoecium, the female gametophyte. It is a female reproductive part. It may have a single pistil, monocarpellary, and if it is more than one, it is called as multicarpellary. So these two are multicarpellary structure. It is a mono single carpel, and this is multicarpellary. Where you can see it is completely fused means it is called syncarpels. If it is carpels are free, they are called apocarpels. This is syncarpus monocarpellary, syncarpus multicarpellary, apo which is free, apocarpus multicarpellary. Now we're going to see stigma, style, and ovarian. What is a stigma? Stigma is a landing platform to receive the pollen grains. It acts like a receptor. A receptor means it will receive the signal. That means when pollen grains land on the stigma. So what is pollination? Transfer of pollen grains from anther to a stigma. Maybe the same plant or same flower, same plant, different flower, different flower, different plant. Three categories are there. About that we could discuss tomorrow in the next class. So we have stigma, style, and the ovary. So stigma is the landing platform for the pollen grains, and through style is elongated part beneath the stigma. What is the tube which will grow in the or which will pass through the style?
Pollen tubes.
Pollen tube will pass through the style. So in stigma, pollen will germinate. In style, pollen tube will enter. And ovary is the basal part, which is bulged part. And of course, inside that only ovule is present. And we have seen the magnified image of an ovule also. And the cavity between the ovarian wall and the ovule is called as your ovarian locule, and the ovary is you can see here is your ovary and this is your ovule is connected to your ovary by means of placenta. So we have learned about placentation in grade 11. Arrangement of ovules on the ovary is called as placentation. We have marginal, axile, free, central, basal. We have learned in your grade 11. Arising from the placenta are the ovules. The ovules may be one, ovules may be many also. Now, structure of megasporangium. So you know that funicle is a part of ovule or a part of ovary. Funicle.
Ovules.
It's a part of no funicle beta. Funicle.
Unique is ovaries, sir.
Yeah. So the ovary and the ovule. So this is the ovule, and we have the ovary. So ovary and ovule are connected by placenta. So funicle is a part of your ovary, or funicle is a part of placenta. So funicle is a small stalk which attached to the placenta. So ovule is attached to the ovary by means of placenta by a stalk called funicle. So what is hilum? Hilum is the midpoint or a junction which is connection between the placenta of the ovary and the ovule. Now there are two integuments, outer integument and inner integument. So when you look about an ovary and ovule, then we can discuss about other parts, what they will transform into. Outer integument is there, inner integument is there, but it is incomplete in one particular area. That means a small tip is there where the integument is not completely enclosed. That area is called as micropylar end or micropyle. Opposite to the micropyle is the chalazal end. How we have in the animal cell, animal pole and vegetable pole. So this is used for identification, micropylar end and the chalazal end, where we have another group of cells here, and it is called as nucellus. There we had tapetum. Tapetum will nourish the microspore mother cell, and the nucellus will nourish the megaspore mother cell located inside the embryo sac. Embryo sac, when they are young, it is seven celled or eight nucleata when they are young. When they are not matured,
8
it
nucleates.
is 8 nucleate. When they get transformed and matured, it becomes 7 cell. About that condition also we have discussed. So when you look up enclosed within the integuments is the nucellus. It contains reserved food material, and inside the embryo sac only the megaspore mother cell is present, which undergoes transformation and mitotic division. As well, first it is meiotic followed by mitotic to form a complete embryo sac. About that only we could discuss now. Now we have the megaspore mother cell. The megaspore mother cell undergo meiotic division number one to form megaspore dyad. Each and every megaspore dyad undergoes meiotic division two to form a megaspore tetrad. So we have got about four megaspore tetrad here, children. There in megasporogenesis, we got four microsporogenesis. We got four microspores, and all were functional. In megasporogenesis, from one megaspore mother cell, we are getting four megaspores. But when you look about the four megaspores, tell me that among the four megaspores, all are functional. Any one is functional.
1,
Only
sir.
one.
Any one is functional. So three of them will get degenerated. Three of them they get degenerated. Only one of them will become functional. Since the matured embryo sac, which is seven celled, actually eight nucleate. From where they are developed, they are not developed from all the four megaspores. All the three have been degenerated. That means among the four, three have been degenerated and one has become functional. That functional megaspore is developing into a seven-celled eight nucleate. Now my question I'm going to shoot before you all. The megaspore mother cell transform into two cell to a two nucleate, then followed by four nucleate and eight nucleate. In this, karyokinesis, cytokinesis takes place together, or only karyokinesis takes place.
Only karyokinase.
Only
Only
karyokinesis.
karyokinase.
Yes, cell wall formation will not take place. That is why I'm not talking about a two-celled, four-celled, eight-celled. I'm not using that. I use a condition called two nucleate, four nucleate, eight nucleate. Then only I'm using a term called seven-celled at only one particular state with maturation. Because only cell wall formation is not taking place, only karyokinesis takes place. So the nucleus of the functional megaspore divides mitotically to form two nucleate embryo sac. Each and every two nucleate embryo sac undergo again mitotic division to form four nucleate embryo sac. Again, and they move towards the opposite pole. Again, they get transformed into eight nucleates. So 1, 2, 3, 4, 5, 6, 7, 8. Eight nucleate are there. Among them, I'll go for the next one because this you won't be drawing in the exam. The drawing you'll be writing in the exam is the next one. Among them, Three of them are grouped in the chalazal and they are called as antipodals. So out of eight, three become the antipodals. So three nucleate, three antipodals. Two of them becomes synergids and one egg cell which is haploid. Then we have a central cell where we have two polar nuclei. Total number of cells, three antipodals, one central cell and one egg cell and two synergids. It is seven celled. But central cell is having two nuclei, that was eight nuclei. I have given you a tabular column in your classwork copy. You could have noted. And with that, if you draw this diagram, if you note that, it is very easy. Now, here a question is then what is egg apparatus? Egg apparatus is equal to two synergids and one egg cell is called egg apparatus. Why, sir, I'm not giving importance for antipodals or all these things? Why I'm calling only this area as an egg apparatus? Here I have some apparatus called filiform apparatus. Here in the diagram of pollen grain which I have given you in pollen germination, the tip we have the transformed vegetative cell. What is that cell called as? Or what is that is called as?
Tube nucleus.
Tube nucleus. The tube nucleus can enter into the antipodals also. They can enter in this way, they can enter in this way, and they can enter in any number of ways. But most of the angiosperms they enter only through the micropylar end. So how they are able to search? Now I'll ask you the question like this to understand scientifically. I'm giving you. See, it's a conceptual information I'm going to give you. For a woman who attained puberty, 1 month left ovary will functional, next month the right ovary will become functional. If the woman is getting married, and of course they're going to have a child, but how the sperms know that left side ovary is functional, right side ovary is functional? Because sperm can carry anywhere. So what will occur? The secretion from the ovary or the released part called as ovum will attract the sperm by some chemicals. Similarly, the filiform apparatus will guide the pollen tube because if they enter into this path, they have to the pollen tube have to go for a long path and they may fuse with chalazal or they may fuse anywhere else. That means it becomes polyembryonic. About the concept I'll be teaching you in the last class. They can enter into synergids also. No. Here we have the filiform apparatus at the synergid. They won't enter into synergids. Through the filiform apparatus in the synergids, they will enter into and reach the X. Therefore, children, when I'm talking about that, what is the role of filiform apparatus mean? It will guide the entry of pollen tube. What is the role of filiform apparatus? Anyone?
Guide
To
entry
guide
of
the
It
entry
will
of
guide the entry of pollen tube. Okay, fine. So now this and all is about various organization. Among the eight nucleate which we have, it is actually seven cells. Three cells are grouped at the micropylar end, constitute the egg apparatus, consist of two synergids and one egg cell in the micropylar end. Micropylar end is the end where integuments are present or integuments are absent.
absent.
Wow, thank you very much. Integuments are absent. And next, when we look about here, okay, they have a cellular, means some filiform apparatus to guide the entry of pollen tube into the synergids. About that, we have seen. Three cells are there at the chalazal end. And what is the role of the chalazal end? In that, we have antipodal. In that, we have antipodals. And apart from it, they have a large one central cell, including the two polar nuclei. Therefore, this is the stages we have here. Three are becoming degenerate. One is functional. They undergo FG1, FG2, FG3, FG4 are simple mitotic division they are enclosing. So, three mitotic division occurs without cellularization, that is, no cell wall formation. So, 2 nucleate, 4 nucleate, 8 nucleate. Fine. Among them, you can see they get organized into 7 cell, 8 nucleate. About that, we have discussed. These two synergids, one X cell, three antipodals, one central cell with a two polar nuclei. Any doubt, children?